Thank you. So thanks very much for being here. I'm the Chief Medical Officer at BIOINVENT. BIOINVENT is an antibody company entirely dedicated to immuno-oncology. The company started a few years ago. It's publicly traded in Stockholm. It started when everybody was doing antibody engineering. Phage display, antibody, etc. So the company created a library that has been proven very successful over the years. We've worked for a number of partners who basically have used our antibody library to get antibodies and do further development in the clinical space. That's just a piece of history, but I will tell you how that has translated into a very interesting recent story. We have a broad portfolio. We did, looking into that platform that I will describe briefly, we did a deal with Pfizer about 3 years ago. They came to the company, looked at the platform, and thought it was interesting. We have been able to deliver, provided to them some targets, etc. So that's very good. We have also a strong institutional shareholder base. Pfizer came in as an investor. Omega Funds, Merieux and Van Herk, a family office in the Netherlands, and Raymond Healthcare, the largest healthcare equity investor in Sweden. And the company has a renewed management team that is moving these things forward. Basically, I'll speak about this. This is the platform that I'm talking about. Basically, what we do, we have a strong relationship with the Lund Hospital in Sweden. We get fresh tissue, tumor tissue from them on a more than weekly basis. We do not do anything to that tissue. We do not culture it or undertake anything. We basically throw the 20 billion antibody library on top of it, do some differential screening where the company has become very competent. The platform is called FIRST. We have very wide patent protection over that library, over that platform. So basically, we cherry pick the antibodies that we think or that we see are having the best activity. This is in a target agnostic manner, so we don't know what the target is. We run all the mouse models in order to understand which ones are the best antibodies that we need to pick. And once we have found the best antibodies, we go back, find the target, and pursue the development. We have been doing this for a number of years now, about 3 years. Pfizer came in when the platform was only conceptually created. They were very happy with the approach, and that's why we signed this deal with them. Obviously, in the immuno-oncology is probably the largest growing area in oncology. It's no secret to anyone. Yet, most of the patients do not respond to immuno-oncology therapies. So there is a huge medical need, which I think, and when we see the presentations here, it should continue to grow in the near future. This is the immunology, the immunity cycle in cancer, where obviously it's very complex. With this platform, we can use it as a huge telescope to look into the tumor microenvironment. That's where basically, and we select what we do. So basically, we look in the Pfizer collaboration, we look at myeloid-derived cells. We're also looking at regulatory T cells, and we are looking at things that inhibit existing antibodies from working. Those are currently 3 areas that we're doing. And that has translated in the past few years in the current pipeline, which focuses in those 3 areas that I mentioned. The first one and most advanced is Fc gamma receptor 2b I will comment a little farther about that because I think that's a super interesting target. We're currently in phase 1, just going over to phase 2. I'll tell you a little bit about what we're doing in regulatory T-cell space and a little bit about what we're doing with tumors, myeloid cells. For the first line there, Fc gamma receptor 2b, the interesting thing is that most of the work that has been done in immuno-oncology has been done around the targets on T cells, which are here depicted on the right. Obviously, CTLA-4, PD-1, PD-L1, that's obviously super important. Mostly the ones that have worked are inhibitory receptors. So when you block the brakes, that's when it works best. That's what we've learned in the past 10 years. Interestingly, the innate branch of the immune response, so macrophages, dendritic cells, et cetera, which are actually the cells that capture whatever needs to be captured and presented to the T cell, to the other arm of the immune system, the adaptive arm of the immune system, that has been not very much exploited. And the reason for that, and interestingly, the receptors there also are similar. You have inhibitory receptors and activating receptors. Interestingly, there is only one single inhibitory receptor. There is only a single brake. That is the target of our first program. You may be asking yourself, why is this so difficult, et cetera? Actually, generating an antibody against Fc gamma receptor 2b is extremely difficult. Fc gamma receptor 2b is 95% identical to Fc gamma receptor 2a. So if you find an antibody, it's extremely difficult to find an antibody that will specifically recognize Fc gamma receptor 2b and not Fc gamma receptor 2a. Throwing those 20 billion antibodies that I mentioned, we were able to find such an antibody that not only recognizes it very specifically, but also blocks it. So there is no possibility of signaling through that receptor. That antibody is in clinical development. We moved it in non-Hodgkin's lymphoma because that was the reason, that was the first data that we had. This is what the antibody does. There is a little bit of a tweak. You may remember that non-Hodgkin's lymphoma is treated with anti-CD20, rituximab, obinutuzumab, et cetera, et cetera. What happens there is that CD20 is recognized by rituximab, but if the tumor cell is expressing Fc gamma receptor 2b, there is that trimer that is pulled into the cell, and you lose CD20 activity from the surface, and the macrophage does not do any of the work that it's supposed to do. What we're trying to do is block Fc gamma receptor 2b, so rituximab, so CD20 stays on the surface, rituximab recognizes it, and then you have phagocytosis and all the engagement of the innate immune system. Just a little bit of data. Here you see those are transgenic mice, double transgenic mice, where you see that if you give the two antibodies separately, you have a little bit of activity, but when you give the two that you have a great deal of activity, and this is actually data from patients who have become resistant to rituximab. You probably know that in most patients that are in non-Hodgkin's lymphoma are treated with rituximab, and they stop responding after a while, and just about everyone stops responding. So then there is other drugs, et cetera, but these are patients, cells from patients who were actually not responding anymore, and in a very short course of the antibody treating these cells, we see that we already get responses because of this mechanism of action. With this hypothesis, we moved into the phase 1/2a trial. We're currently in part A of the trial. This is, we're taking people who are resistant to rituximab, who have received at least one line of rituximab in the past. We're treating them with the two antibodies, and then we're moving, and then we're continuing the treatment. Right now, we're in the escalation phase. We need to find the recommended dose for BI-1206, that's the name of our antibody. And then we will move into Part B, which is basically an expansion cohort. The interest of this approach is that in this particular indication, we obtained orphan drug designation. Our idea is to obtain breakthrough therapy designation and eventually a conditional approval because it's a small indications. Patients with mantle cell lymphoma, which is one form of non-Hodgkin lymphoma, once they've gone through the several lines of treatment, there is no options and they may die very, very quickly afterwards. So this is the approach that we're taking here. One interesting thing that I should mention is that mantle cell lymphoma patients are high expressors of our target, and that high expression has been associated with poor prognosis. So that's something interesting. There was, in the past 3 years, there was a lot of publications in the best journals in the world from different groups showing that those Fc gamma receptors also modulate the activity of immune checkpoint inhibitors, PD-1, PD-L1, anti-CTLA-4. So that the data was so strong also from our group that we decided to move into a solid tumor space with this antibody. So this trial has been already approved by FDA and European authorities, and we're moving into this as we speak. And finally, one other another antibody that came out of our screen was an antibody against CTLA-4, which we compared with ipilimumab. But the interest of this antibody is that it's a highly deleter, much more strong deleter of regulatory T cells. So we're running this program in collaboration with Transgen in France, and we basically put the antibody into their oncolytic viral system. The data are super interesting. KOLs are extremely engaged. This is something also that we're moving forward. So this is, again, I think I've been a little bit. Finally, one word about another target. You see it there in the middle, 1808. This is an emerging target. There was a paper in Science a couple of weeks ago. TNF receptor 2, we have a very powerful antibody and an antibody program, actually. This is super interesting. That also will be going into clinical development during the first half of next year. So I don't have a lot of time, so these are our milestones. This presentation is on our website, so just a word to say that because of the history of the company, we have cell line development and manufacturing capacity for clinical grade material. So we can move very quickly once we have found an interesting antibody into clinical development. This is the board. Martin Welschof is our CEO. He joined the company about a little over a year ago. We've been doing a number of road shows with him. Bjorn Frendeus, who is the person who designed the antibody and the platform to identifying these targets and antibody. Myself, I joined 2 years ago to move these things into clinical development. Stefan Eriksson, who is our Chief Financial Officer, and Christopher, who leads the manufacturing facility. And we have a top-notch scientific advisory board and also medical advisory board. So we are extremely happy, and it's really big time. This is the board members. Leonard Krumer came about a year ago. He was the chief financial officer of Crucell, who was a nice success story when they were bought by J&J. And in the interest of time, I'll stop here and take 1 minute of questions if there is any.